Hello, welcome to my channel Medium Medicom Medical. Today topic is Candida albicans. Let's study about introduction, pathogenesis, lab diagnosis, test for identification of species, treatment. Let's start. Introduction. Candida is an opportunistic meiosis. It is a yeast-like fungus produced pseudohype. It is most common fungal disease in human and affecting the mucosal skin and various internal organs. Let's discuss about pathogenesis. Candida is a species of part of normal flora of the skin mucosa including gut flora. In the presence of opportunistic condition, they can cause various infections such as mucosal candidas, chronic mucocutaneous candidas, cutaneous candidas, invasive candidas. Let's study about mucosal candidas, oropharyngeal candidas, oral thrush, candidal vulvovaginitis, balanotitis, balanopathitis, and esophageal candidas. Chronic mucocutaneous candidas among infants and children with a deficient cell-mediated immunity. Cutaneous candidiasis, intertigo, erythema, and fistulas in skin folds, franchia and onychomycosis, fungal infection in nails. Invasive candidiasis, results of hematogenous or local spread fungal infection. Various forms are urinary tract infection, pulmonary candidiasis, septicemia, meningitis, keratoconjunctivitis, and endophthalmitis, disseminated candidiasis, and nosocomial candidiasis. Let's discuss about laboratory diagnosis. Specimen collection. Depending on the site of infection, various specimens can be collected such as whitish patches from mucous membrane, skin and nail scraping, sputum, urine or blood. Direct microscopy. Gram staining reveals gram-positive oval budding yeast cells 4 to 6 micrometer size with a pseudohype culture. Specimen can be inoculated on SDA with antibiotic supplements and then incubated at 37 degrees Celsius. Candida can also grow on blood agar. Blood for culture can be inoculated into a blood culture bottle. Colonies appears in 1 to 2 days and described as a creamy white smooth and pastry with a typical yeasty odor. Gram staining of the colonies shows gram-positive budding yeast cells with a pseudohype. Test for species identification. Candida albicans can be identified and differentiated from other Candida species by following methods. CHRO magar different Candida species produce different colored colonies and on CHRO magar Candida albicans produce light green colored colonies. Germ tube test. It is also called as a Reynolds Barude phenomenon specific test for Candida albicans. Colonies are mixed with a human or sheep serum and incubated for 2 hours. Wet mount preparation is examined under microscope. Germ tubes are formed described as a long tube-like projection extending from the yeast cells. It is differentiated from pseudohype as there is no constriction at the origin. Dalmy plate culture. Culture on corn meal agar can provide clue for species identification. Candida albicans produce thick-walled Chymalospores growth at 45 degrees Celsius. It differentiates Candida albicans grows well from Candida dublinesis does not grow at 45 degrees Celsius. Sugar fermentation test and sugar assimilation test can differentiate between various Candida species. Molecular methods such as polymer chain reaction using a species specific primers and useful for species identification. Treatment. The antifungal drugs recommended depend upon their type of candida. Cutaneous candida drug of choice is topical esophageal or ulovaginalis candidas drug of choice is oral fluconazole. Disseminated candidas drug of choice is ampicillin B. Candida glabrata and C. cerevisiae exhibited intrinsic resistance to azoles and are refractory to treatment with azoles. Thank you for watching. I hope you all understood. Like, share, subscribe.